finishing up transcription and then starting translation. And the messages that I hope that you get from these two processes are that they are indeed fairly intricate and elaborate, especially transcription in eukaryotes. It's not quite as elaborate in prokaryotes. But translation is very elaborate in both systems. And fortunately for both systems, the translational process is not that different. So there's fewer things to distinguish prokaryotic and eukaryotic. When I finished last time, I'd finished talking about tRNAs. And what I want to do now is turn our attention to talking about messenger RNAs. And I'll remind you that all of the things I'm talking about are, again, in eukaryotic cells. Eukaryotic transcription is considerably different than prokaryotic transcription, as we shall see, largely because of the fact that RNAs in eukaryotic cells are edited, spliced, and pieces are put together. We don't see that phenomenon happening, for the most part, in prokaryotic cells. Most of the place where people talk about that phenomenon, the phenomenon is called splicing. They talk about with respect to messenger RNAs. But the process actually occurs in transfer RNAs and also ribosomal RNAs, and I'll say more about that at that time. Right now I want to focus on making messenger RNAs. And of course, messenger RNAs are made in eukaryotic cells by RNA polymerase II. And we've seen a little bit about the different promoters involved in RNA polymerase II, so I won't go back and say more about that. But messenger RNAs in eukaryotes look different than messenger RNAs in prokaryotes. And there are two major features that help to distinguish them structurally from messenger RNAs in prokaryotic cells. In eukaryotic cells, what we see is at the 5' end of the messenger RNA, there is a structure that's not found in prokaryotic messenger RNAs. That structure is called a cap. It's called a cap, and that cap arises from the addition of an unusual guanine nucleotide that's added to the 5' end of the messenger RNA. And it's added in a very unusual bond. It's added in a 5' to 5' bond. Why do something odd like that? Well, it turns out there's a couple of reasons that cells do something odd like that. The primary, well, not primary, the two reasons are, first of all, I told you that messenger RNAs are unstable. Cells break them down. They have nucleases that break them down. They don't want to keep messenger RNAs around for a long time. Okay? The breakdown that occurs, especially in eukaryotic cells, happens mostly starting from the 3' end. Nucleases can't touch that 5' to 5' bond. That means that the 5' end of the messenger RNA is the stablest part of the entire messenger RNA, the least likely to be degraded. Well, since that's where the protein starts, the protein coding starts, is near the 5' end, that means there's a good chance that the cell will get the maximum amount of translation out of that sequence if possible. Okay? The other reason that the cap is important is, as we'll see in translation, the cap is actually recognized by the eukaryotic ribosome. So eukaryotic ribosomes look for and recognize that cap. And so that cap may be providing an identity to the ribosome to say, hey, look, I'm a messenger RNA. Translate me. Okay? So that's what the cap does. Now the cap has, as I say, a modified guanine nucleotide. It's got some methyl groups on there. And there are also methylated bases on the internal to that cap. Okay? We're not going to worry about those structures, but suffice it to say that the 5' end is modified both by putting on a guanine and some methylations that occur. All those things together are what we call the cap. Okay, the other thing that we see in eukaryotic messenger RNAs that's different from prokaryotic messenger RNAs is that it occurs at the 3' end. And at the 3' end of eukaryotic messenger RNAs, we see something odd. It's called a poly-A tail. And the poly-A tail, as its name suggests, is a long string of adenine nucleotides, adenine-containing nucleotides. Okay? The poly-A tail is put on after RNA polymerase has gone through a sequence that says AAUAAA. So when the RNA polymerase has made that sequence and continues, shortly after termination of the transcription happens. Coupled with that termination may be the process that you see right here. That is, a cut occurs with adjacent, I'm sorry, not in this sequence, but adjacent to this sequence. Within a few nucleotides, this guy is cut. And then the A's are added by a polymerase known as poly-A polymerase. These A's may be 200 to 300 nucleotides long. Talk about wasting energy. Cells don't waste energy for no reason whatsoever. And one of the reasons they do it, I've already told you. And that is that when cells degrade messenger RNAs, they start at the 3' end and they move inwards. So what this poly-A tail provides is a sort of a buffer, and if you want to think of it more as a fuse, okay, for a firecracker that's going to go off, kind of like the telomere we talked about, that would be accurate. The longer the poly-A tail, the stabler, that is, the longer the messenger RNA will be around, and the cell will be able to translate it. The shorter the tail, the less time it will be around, and the less translation will happen. This provides the cell with another way of controlling how much protein it's going, of a given protein it's going to make. So the poly-A tail provides stability for the messenger RNA. Yes, sir?
Do you think it could have been like a precursor to telomeres, where it's kind of the same thing? Where if you...
Do I think it's a precursor to telomeres? No, actually, I think it's pretty independent of that. The telomeres are there for a very different kind of a reason. And the analogy, I mean, the metaphor I've used of the fuse holds for the two. But I think it's for a very different reason that that's there. Yes, sir?
Is there a specific enzyme that breaks down the poly-A tail, or is it just...
Is there a specific enzyme that breaks down the poly-A tail? Good question. No. They are exonucleases that generally start chewing in. They don't go very far, so they chew in a little bit each time. But there's a variety of exonucleases that will do that. Now, there's a second reason why the poly-A tail is important, and it turns out the poly-A tail is also used in translation. It's a recognition factor for the ribosome. We'll see that when we talk about eukaryotic translation. So the poly-A tail is useful for that. Now, interestingly, and it's not completely understood, interestingly, there are some messenger RNAs that are made that don't get poly-A's put onto them. So it's not an absolute thing. The vast majority do, but not every single messenger RNA gets poly-A put onto it. And you might suspect that they're less long-lived messenger RNAs. In some cases that may be true, in other cases it may not be, and as I say, that's an area of investigation. Okay, so that's how structurally the messenger RNAs differ. There are, as I said earlier, some things that happen to eukaryotic RNAs in general. Now I want you to think transfer, ribosomal, messenger RNAs. Some things that happen to these RNAs in general that don't happen in prokaryotic cells. Now I will say one thing first that is similar to what happened in prokaryotic cells, and that's chemical modification. We see chemical modification occurring to these RNAs in both prokaryotic and eukaryotic cells. So chemical modification occurs in both. But the things I'm getting ready to talk to you about do not occur in both. One is an interesting phenomenon known as RNA editing. And RNA editing is, when you first hear about it, you think, why in the world does the cell do this? Well, you know why it does it, but you wonder how it ever came about. RNA editing, here's a real good example of RNA editing. This is a process that occurs for a protein that you've already heard something about. I just didn't give the protein a name. When we talked about chylomicrons and we talked about LDLs, I talked about apolipoprotein complexes. And these complexes were involved in carrying the fat and fatty acid and vitamins and cholesterol within them. Okay? And it turns out that there's, in this particular case, an apolipoprotein known as Apo-B100 or Apo-B48. Okay? The same gene in the DNA codes for both of these. Okay? So if we look at the DNA, same gene. So how do we get both or two different proteins out of the same gene? Well, this happens as a result of a different kind of chemical modification that happens to the messenger RNA. When this gene is transcribed as a messenger RNA in liver cells, the liver cell goes along and it translates that messenger RNA, and it goes all the way through, and it gives a protein that's about 100,000 kilodaltons in length, which is why it's called B100. And it makes this lipoprotein that appears in the LDLs. It appears in the LDLs as seen on the top. And you'll see it has two domains. It has a place to bind the fats and fatty acids and fat-soluble vitamins and cholesterol. And it has another portion that binds to the LDL receptor. Remember how LDLs are carrying things into the cells, and they bind to that receptor through receptor-mediated endocytosis. This is a handle for that receptor, so they can bind to those cells and deliver these goodies. However, if we are in the intestinal mucosa, where the chylomicrons are being made, an enzyme is present that is not present in the liver. The enzyme is a deaminase. It removes an amine group from a very specific cytosine internal to the messenger RNA. When it takes that amine group off, the C is converted to a U. Very simple change, but a very big change. Because when we do translation, UAA says stop translation. So we've converted something that's not a stop translation signal to something that is a stop translation signal. So in the intestinal mucosa, where the chylomicron proteins are being made, a protein is made that only goes through the first 48 kilodalton, same thing, size protein. So we get a truncated version of the protein in chylomicrons. Now, exactly the same gene in the DNA. However, this guy gives this, the chylomicrons get this protein, and of course chylomicrons don't get pulled into the cell by receptor-mediated endocytosis. So a very simple change has very big and very different effects on the protein as well as the complexes that use that protein. Question about that? Jenny?
Does this deaminase go around to lots of different messenger RNAs?
Does this deaminase go around to lots of different messenger RNAs? And the answer is no. That's why I say you wonder, why in the world? You know? But it's there because it works. It's interesting when we look in other organisms, especially lower on the evolutionary scale. Trypanosomes, for example, involved in causing malaria, do some very bizarre types of editing. And they don't just change the sequence like this. They actually end up inserting U's. They put a whole bunch of U's in a whole bunch of genes. And you think, why didn't the DNA have this in the first place? You know, it doesn't seem very efficient. It works for them, but it doesn't seem very efficient in terms of how that's set up. This is an odd process, and I won't say this is the absolutely only thing it works on, but it's not commonly recognizing a bunch of other factors. You can imagine it's recognizing some sequences and knowing where it has to do that deamination. Okay. So that is editing. Now, a super type of editing that occurs is a related thing, but it's not so much a chemical modification as we saw in the case of the Apo-B100, as much as it is a mixing and matching of segments of RNAs in a phenomenon known as splicing. This is what I referred to at the beginning. Splicing is a process whereby portions of an RNA are removed and the pieces that are left are joined together. That's the splicing part. So here is a schematic of what's called a splice junction. Okay? So the pieces that get removed from an RNA during splicing are called introns. They get removed and they disappear. They get chewed up later, and I'll show you that. Okay? But they disappear. The mature RNA That is the one that's had the introns removed from it. The mature RNA contains the remaining sequences, and they're called exons. Now, splicing occurs in almost every, again, not absolutely every, but almost every eukaryotic RNA, or messenger RNA, certainly. Not so much ribosomal and transfer, but almost every messenger RNA gets spliced. The splicing may involve removal of a single intron. It can remove as many as 50 introns, depending upon the gene. Okay? Now, when splicing was first discovered, people said, how in the world? This can't be very common, number one. This can't be very useful, number two. But it turns out to be very useful, and I'll give you some implications about that in a minute. But first I want to talk about how the process occurs. There appear to be some good evolutionary reasons for why splicing occurs. When we look at the splicing, we'd like to say, well, we'd sure like to know if we have a given sequence of nucleotides in a messenger RNA, we would sure like to know where in that sequence the introns are. We'd like to be able to say, okay, well, here's a sequence that I can find in this intron, and therefore, or I can see in this messenger RNA, so this portion must be an intron. And that's been a bit puzzling. What you see on the screen are some sequences that are commonly found, but not absolutely found in introns, meaning that there are some introns that violate the rules. The only sequences that are absolutely found in every intron are at the 5' end of the intron, there's a GU. GU are terrific, right? At the 3' end of the intron, there is always an AG. Commonly, you see these other nucleotides around it, but not always. One of the mysteries of splicing is how does the cell know which GU to get? Because there's a lot of GUs in a sequence, and not all of them get removed as introns. It's important the cell get it right, because if it doesn't get it right, it's going to make a messenger RNA that's not going to have the right sequence of amino acids for making the protein that it wants. That's a problem. Okay? The other thing that we see within the intron is we see a region that has a decent number of pyrimidine nucleotides adjacent to an A. And that A, we'll see, plays a role in the splicing of the intron. Okay, so those are the common, and there's not very many, but those are the common structural features we see of an intron-exon junction, a splice junction. Well, let's look to see how this process occurs. This is a terrible figure, by the way. Okay, coming from somebody who can't draw, a stick figure, that says a lot, I think. But this is a bad figure. Here is the messenger RNA that has been made by the RNA polymerase. Remember that when it's first made, it's going to have the introns and everything in it. Okay? In this process, the first thing that happens is there's a nucleophilic attack. Here's that A next to the pyrimidine residues I told you about. There's a nucleophilic attack by the A on the phosphate that's right next to the GU at the 5' end of the intron. So this guy makes an attack right here, and that frees the exon from this. In other words, this bond between here and here is broken as a result of this attack. That causes this guy, as it shows here, it looks like it makes a gigantic move, which it really doesn't, but it makes a flip so that now the 3' hydroxyl is free, and it attacks the phosphate at the junction of the next exon, so that the first exon becomes joined to the second exon. Okay? So we've taken something that went here and linked it to there. Now, as a result, exon 1 is joined to exon 2, and in the process, what flips out is something called a lariat structure. A lariat structure, okay? It's called a lariat structure because it has, like a lariat does, an end of a rope, and it has a loop. This guy will be degraded by nucleases inside of the cell. Degraded by nucleases inside of the cell. Now, there's something that happens here that we don't see happening anywhere else in the cell. In the cap, for example, when I talked about the capping of the messenger RNA, we saw a bond we didn't see anywhere else. That was a 5' to 5' bond. Haven't seen that before. You're not going to see it again. It's an unusual bond. This guy has an unusual bond. It's a 5' to 2' bond. And you might say, well, why does it bond to the 2' end? Because the 3' end is bound to the phosphate. The 2' is the only hydroxyl on that ribose that's available for the bond. So this makes a covalent bond. This lariat gets flagged as something for destruction. Nucleases come along and chew it up. Meanwhile, this guy is ready. If this is the only splice site that's in there, this guy is ready to go out of the nucleus and be translated. Or if it's a ribosomal RNA, to go out of the nucleus and go join a ribosome or something. Okay? Questions on that? I'll say a little bit more about mechanism later also on this. Everybody's ready for the day to be over? I am too. All right. There's that 5' to 2' bond. Okay? There's the guanine that was at the 5' end of the intron. There's the adenine that was at that branch point next to the pyrimidines. There's the 2', 5' bond. Here is the rest of the lariat structure, that handle of the lariat, coming down over this way. Yes, Megan.
So is this process free of enzymes then?
Is this process free of enzymes? No. In fact, this process is full of enzymes, and that's why I'll talk about that later when I start talking about the proteins involved in this. Interestingly enough, when we look in, as I said, splicing doesn't occur in bacteria. There's one exception, or one or two very minor exceptions. When we look in bacterial viruses, called bacteriophages, there's at least 2 places where there are messenger RNAs that are known in bacterial viruses where the splicing occurs all by itself. No proteins involved. So that's a kind of a cool thing. That was actually where the catalytic RNAs were first discovered, when somebody discovered that an RNA could splice itself. Kind of cool. Okay. Now, the question about proteins involved, yes, as almost anything else that happens inside of cells occurs, there are enzymes and proteins and RNAs that all help the splicing process to occur. Notice I said proteins, which are enzymes, of course, and RNAs are involved. What you see up here is a listing of the proteins. They're called snRNPs, which stands for small nuclear ribonucleic proteins. Come here with the small nuclear ribonucleic proteins. Okay? Nucleoproteins. All right, close enough. These guys are proteins that facilitate that splicing process. They are assisted by small nuclear RNAs, snRNAs, that have similar names. In fact, these guys generally carry an snRNA. U1 snRNP contains the U1 snRNA. Okay? Now there's a complex of these that comes together to facilitate that splicing reaction. I'll just point out a couple functions here. U1 binds the 5' splice site. We'll see this in the mechanism. That it's helped by the U1 snRNA. U2 snRNP binds the branch site and forms a part of the catalytic center. The branch site is that A nucleotide that's next to the pyrimidines that I talked about. Okay. Here is a close-up of the U1 snRNA. This guy is in the U1 snRNP. And if we look at its sequence, look at that. Complementary to the 5' intron. This guy is going to play a role in aligning the U1 snRNA with the 5' junction of the intron. This is one of the ways in which the cell is learning where that intron is and where to start doing its cutting. And notice this says mRNA precursor. This could be a ribosomal RNA. This could be a transfer RNA. Your book, like many books, describes this as an mRNA process, but it's not exclusively that. Okay. Now, you'll notice that the title of this slide was Spliceosome Assembly. A spliceosome is a complex of these proteins, the snRNPs, the RNA that's being spliced, and the snRNAs. This complex is called a spliceosome. The spliceosome has to assemble in order for the splicing to occur. This is, as you could imagine, a fairly orchestrated process. The first thing that comes in to start the assembly of the spliceosome is U1 snRNP, assisted by that U1 snRNA making those pairings. U2 snRNP comes in second. Then the rest of the spliceosome assembles. We're not going to worry about the details of that. Okay? And the bonds that I described before to you are all broken and made. That's where we had that nucleophilic attack of the G onto that phosphate at the junction of the intron exon. I'm sorry, not G. It was an A. Sorry, I said G. Where the A attacks that. Okay? All those other parts of the mechanism I talked about are all happening inside of this spliceosome. This shows a little bit more clearly, although it's kind of hard to get your head around the whole thing that's happening here, but this shows a little bit more clearly how all these sequences are in fact arranged with each other. And one of the things that we see that I think is very interesting is if we look at this exon intron junction here, okay, what we see is here's that A that was in that pyrimidine-rich sequence, and look how the base pairing causes it to bulge out. This thing's sticking out like a sore thumb. And that sore thumb is pointed, thanks to these proteins orienting everything, that sore thumb is pointed right at this junction. It's this bond that breaks this one that frees this end to join over here. Okay? Pretty cool stuff. Pretty cool orientation of things that are there. Okay. Okay, let me stop there and take questions on that mechanism I just talked about. Okay. All right. There are diseases that are known that result from problems in splicing. Splicing has to be pretty precise because we're messing with sequences that are going to be translated. If we don't splice it properly, we're going to put ends together that shouldn't be put together, or we're going to miss a nucleotide or two, and that's going to totally screw up the translation of that if it's a messenger RNA. So splicing needs to be precise. If splicing is not precise, or there is something that affects the splicing in a negative way, we can imagine that the genes that are translated from that might have some problems. And they do. Okay? One of these is a set of blood diseases known as thalassemias. The root cause of thalassemia, some of the thalassemias at least, is a change by mutation within an intron. That change inside the intron causes an AU to become a GU. And GU, you may remember, what is just like the GU we saw over here. If this GU is recognized by the splicing machinery as the 5' end of the intron instead of this GU being recognized as the 5' part of the intron, then the messenger RNA that's made from it now contains the wrong sequence. Here's the properly spliced one. Here's the improperly spliced one. The improperly spliced one is fine until you hit the stop codon. The person that has this mutation will not make a fully functional globin gene. The thalassemias affect the globin genes of the blood. Okay? These can be very severe, as you could imagine, and this is going to be a problem. This guy may ultimately be degraded much faster because it's not being used for translation. There are many other examples of mutations affecting disease. Now, one of the questions people have is, well, why go to something as elaborate as splicing? Why cut out all those nucleotides that cost all that energy to put in the first place? Why don't you just have it like bacteria do? Bacteria just start, and they go for a long stretch. Nothing interrupting them, nothing having to take out. You just translate it, you got it. Why are eukaryotes so much more complicated? And the answer is because eukaryotes are so much more complicated. Okay? Remember that we have to differentiate. We have to have proteins that do different things in different cells. When people first started looking at the size of the human genome and estimating how many gene sequences that there were inside of a eukaryotic cell, how many genes would it take to make a eukaryotic cell compared to, say, a bacterial cell, the estimate was that it would probably take 150,000 to 200,000 genes to make a eukaryotic cell compared to a bacterium which has, say, 3,000. Okay? When they completed the sequence of the human genome, what they found was there were about 30,000. One-fifth or one-sixth of what they expected would be needed to make a eukaryotic cell. Well, how does the eukaryotic cell get away with it? One of the ways it gets away with it is by splicing differently in different cells. That means that a cell can take the same gene and mix and match exons together depending upon the needs of the cell. That's a pretty cool thing. So here is an example of a calcium binding protein. In neuronal cells, it looks like this. In thyroid cells, it looks like this. Same starting gene, just mixing and matching of the exons to make related proteins with individual functions for the different cells. This phenomenon, as we'll see when I talk later about the immune system, gives rise to tremendous numbers of possible mixes and matches, and it's one of the things that gives our immune system the diversity that it has. Okay? We have the ability to make something like 10 to the 12th different antibodies. Well, that's a very impressive number, except for the fact we've only got 10 to the 9th nucleotides in our genome. That means we have 1,000 different antibodies for every nucleotide of the genome. How do we do that? We do it by mixing and matching. So this mixing and matching gives us tremendous versatility, gives us tremendous diversity, and we could imagine that this mixing and matching that I'm talking about here would be tissue specific. I'm going to get one type of mixing and matching in neuronal cells, a different type of mixing and matching in in thyroid cells. Yes?
Does this mixing and matching make it so that a mistake in DNA replication is even more serious than like bacterial cells?
It can be. And so his question is, can the mixing and matching make a mutation more of a problem? It can be, but it can also go the other way. So it's not an absolute thing. In the case of the thalassemia, yes. In fact, that's exactly what happened. You saw a mutation that was happening that wasn't even in a protein coding region. It was in an intron. And introns, for the most part, are throwaway sequences, right? Splice them out, throw them away, you chew it up. There was a place where a mutation that had a greater effect because of its location. Yes?
So if you're only slicing out the intron, how is that changing the expression of the genes moving exons from certain promoters so that they get expressed?
No. It's not changing expression because, remember, expression has already happened. The RNA is there because the promoter has been recognized and the RNA polymerase has come along. Okay? So it's not moving it closer to the promoter because, remember, the promoter's in the DNA, not in the RNA. So what it's doing is it's pairing different exons together that do different things. One of the things that we see, oh yeah.
Is it actually removing exons with the intron?
It may. It may. It's a very good question. Is it removing exons? So if we look at this guy here, here is 1, 2, 3, 4. We've got the red sequence here. This splicing has occurred 1, 2, 3, 5, 6. It's actually removed this as part of this big intron. So in some cases, it may remove an exon in that process as well. One thing it will not do is it will not shuffle. It will not take brown, blue, brown, red, brown, blue. It can't shuffle. That's not possible. They have to be sequential. So I might see brown, 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 blue, blue, like I see here. I might see brown, brown, red, blue, but I could not see brown, red, brown, blue. Make sense? Okay. Now, this gives tremendous diversity. I want to emphasize that. I'll come back and say more about that when I talk about the immune system. Now, the last thing I'll mention was the question that I think was Megan asked a little bit ago about, well, does this all occur in the presence of proteins? And in eukaryotic cells, the answer is generally yes. Not absolutely, but generally yes. There are some sequences that are known in eukaryotic cells that are self-splicing. A molecule that's reacting within itself to cut a piece of itself out, okay, and join the remaining pieces together. That's what you see on the screen right here. It's a process that in its mechanism doesn't look significantly different than we saw with the protein catalyzed reaction. The result was the same, though. We still get the exons joined together properly. This phenomenon is known as self-splicing. Self-splicing occurs when cells have something built into the structure of the RNA that facilitates this happening. This process, when it was discovered, it ultimately resulted in the winning of a Nobel Prize by the person who discovered it, okay, demonstrated for the first time that RNAs can be catalytic. They can catalyze reactions. Prior to that time, the only things we thought of inside of cells that catalyze reactions were proteins in the form of enzymes. So a new term was created to describe this process. These guys became known as ribozymes. A ribozyme is a catalytic RNA. There are other ribozymes that are known, including one I'll talk about next week in translation. Ribozymes can work on themselves, as these guys do, or different ribozymes can work on other things, as happens in translation. Yes?
What's the rate of ribosomes versus mRNA?
The frequency with which you see the self versus the other. The self is pretty rare. The self is not very common, the self-splicing. I know of maybe there's probably maybe 30 or 40 examples, I would guess, of self-splicing introns that are known, self-splicing RNAs that are known. Everything else, thousands, millions, are all catalyzed by protein. Now, one exception, like I said, is the fact that there's a bacteriophage that actually does self-splicing. That's the only place in the entire non-eukaryotic world where splicing is known. The only place in the non-eukaryotic world where splicing is known. You can call a virus a prokaryote or whatever you call it, a virus. I don't know what you want to call it, but it's definitely not a eukaryote, and that's the one place where we know that it occurs. Okay, so that's the splicing story. Yes? Okay, yeah, it's a good question. So are these things found in a genre of proteins? I don't know of any specific patterns associated with them. No. The place where Tom Cech discovered them, the person who won the Nobel Prize for them, it wasn't even in a messenger RNA. It was in a ribosomal RNA. So I think it's more a function of how that individual sequence itself evolved than it is the protein. It's a very good question, but I don't know of any pattern with, let's say, this is a protein that's involved in, let's say, DNA replication or something, and this is where we see it. There's nothing like that, no. Yes, Jenny?
Going back a little to the tissue-specific splicing
Yep.
for different proteins, is that done because the cells express different spliceosomes, or how do they...
Okay, so is the tissue-specific splicing enabled by different spliceosomes, I think is what you're saying, right? So one tissue does it one way, the other tissue does it the other way. And the answer is yes, there are differences in the U1, U2 RNAs that recognize things, as well as the proteins that are involved in that. And as you can imagine, that's very actively studied. Differential splicing provides control to cells. Not only does it provide diversity, but it provides a way of controlling which protein is made and how much of it is made. So in some cells, you may see two different splicing forms of the same protein. How much of one versus the other depends upon how efficiently one splicing is done versus the other splicing. So there's some very interesting controls that happen with that. Yes? Yeah, well, this is a precursor of forming the lariat structure. When we look at the self-splicing mechanisms, they're similar to what we see in the lariat structures, and they're just simply closing the covalent bond in doing that right there. I don't know, I can't tell you why, mechanistically, that is just the way that it does occur. But this is one, in fact, let me just show you the next figure with this. You'll see some interesting relations. There are two different classes of self-splicing RNAs that are known. And right here. Group 1 and Group 2 are both known as self-splicing introns. And when we look at Group 2, it's very similar to the protein catalyzed one, much more similar than the Group 1 is. And we see, for example, in the Group 2 self-splicers that we actually do see a lariat structure, and we don't see that over here with this structure. So there are some differences mechanistically with them. But you can almost see an evolutionary progression in going from this to ultimately something that was catalyzed in assistance with protein. Yes, Megan, last question.
So when the cell is using like the essential amino acids, do they ever combine with the intron pieces to make certain proteins, or are they completely separate proteins that are made by essential?
Okay, so essential amino acids really has nothing to do with this process. So essential amino acids has to do with, when we call something an essential amino acid, it means do we have to have it in our diet compared to do we make it. But in terms of function, there's no difference between them.
Okay, I just was wondering if some of these amino acids pair with some of the essential amino acids to make certain proteins.
No, no. Okay. Let me move on then, and we'll start translation. And now I've got about 5 minutes to talk about that. That'll give us a decent introduction to the topic. And we're off and running. Translation. Everybody's heard about it in basic biology. You know something about it. Translation is the process where we convert nucleic acid sequence information to protein amino acid sequence. We convert nucleotide sequence information into amino acid sequence information. That's what translation's about. That process occurs in ribosomes, and it requires all three of the major types of RNA that I've talked about. The ribosomal RNA, the messenger RNA, and the transfer RNA. Okay? The ribosome is a complex of proteins and RNA that facilitate this happening. The ribosome looks something like I like to describe as a snowman structure. It has what's called a large subunit, and it has what's called a small subunit. This doesn't look very different in size, but the large subunit is on top, the small subunit is on the bottom. We know the structure of the ribosome due to some amazing X-ray crystallography work that have given us some tremendous insights into the mechanism of translation. The structure of the ribosome now has been known for a few years thanks to, as I said, some amazing X-ray crystallographic work. When we look at it, what we see, as I said, is what's called a large subunit that contains 30 or 40 proteins. It contains some ribosomal RNAs. And a small subunit that contains about 20 or 30 proteins and also some ribosomal RNAs. The messenger RNA feeds in between these two subunits, and transfer RNAs come in carrying amino acids to be joined together to make the protein. They come in and make base pairs with sequences in the messenger RNA, and the place where they come into the ribosome is known as the A site. They come into the A site, and everything happens in these three sites for translation: the A, the P, and the E. Okay? And I'll say more about those when I talk about mechanism next time. as the protein is being made, it comes out of the large subunit, as you see at the top. It comes squirting out the top through a little hole. And once it's out here, it's free in the aqueous medium, in the cytoplasm, and it starts to fold. So folding can start as the protein is coming out of the ribosome. In some cases, it may not occur until later, but it can actually start folding as it's coming out and as it's being made. Okay, translation, like transcription and like DNA replication, occurs exclusively in the 5' to 3' direction. Exclusively in the 5' to 3' direction. You may remember last term we talked about the structure of proteins. We said that we had one end that was called the amino end and the other end that was called the carboxyl end. 5' to 3' corresponds to amino to carboxyl, meaning that the first amino acid that's put into a growing polypeptide chain is the amino terminus. The last one that gets put into the growing polypeptide chain is the carboxyl terminus. This shows how the individual amino acids are joined. Here is the first transfer RNA with an amino acid attached to it. At the ribosome, the amino acid on a second transfer RNA comes in, and the amino acid on the first one becomes linked to the second one. Okay? So here's what we see. Here's the second one. Here's the first one. Here's the second one on the first one. This is now the amino terminus. Currently, this is the carboxyl terminus. If translation continues, then a third one comes in, and now the second one becomes joined to the third one. So that we see the carrier of the growing polypeptide chain is the latest tRNA that comes in. Okay? Questions on that? All righty. One last word about translational accuracy, and then we'll finish. Accuracy is something we worry about a lot. Remember, we worried about accuracy with respect to DNA replication. We talked about accuracy in transcription. We said it wasn't quite as big of a deal. And now we're talking about accuracy in translation. How important is it for translation to occur precisely? Well, we want it to occur reasonably well. Otherwise, we're going to make the wrong protein. That's not going to be of much use. If the cell has to make, let's say, 50 copies of a protein to get one that's perfect, that's not very good. So there has to be some reasonable amount of accuracy in translating the code. It turns out that cells are accurate to the limit of about one mistake in every 10,000. About one mistake in every 10,000. And this works out very nicely if we look and say, well, if we have an error of 1 in 100, what's the likelihood we're going to have an error-free protein of 100 amino acids long if we have that high of an error rate? Well, if we have one error in 100, only 36% of those are going to be perfect. If we have one error in 1,000, about 90% of those will be perfect. If we have one error in 10,000, 99% of the proteins of 100 amino acids long will be perfect. That's not bad. Toyota doesn't have that good of a record. Okay? If we're 300 long, 97%. If we're 1,000 long, still 90% of them are good. Accuracy costs energy. The more energy you have to put into being accurate, the less energy you have for other things. So there has to be a balance between having perfect sequences and what you can live with. The cell is willing to live with an error of about 1 in 10,000. If we go to 1 in 100,000, we can get 99% here, but at a very high energetic cost. This doesn't make sense. Okay, good place to stop. Let you guys go and see you on Monday. Yes, sir. How's it going, Stephen?
About interview class next year.
Yeah.
It would be putting 2 more credits on my 60 credit load. We're out of state, so I don't think I should be able to do it.
Yeah,
but
sure.
Do you allow specific classes? I don't know what the policy is.
I can't do that because the Honors College won't allow it. The Honors College has a policy that these are the fixed sizes, and we can't go beyond that. So.
No, I understand. What about like course information? Do you put up the website or anything?
Well, it turns out there's no textbook that's used for the course.
Yeah, I figured that.
And it's all things that I put together. So we don't really have a website. The only thing I do on the website is I post the videos of the students who are doing things, and I don't publicize it outside the class because you don't want everybody else looking at your video, for example. So unfortunately, I don't. Yeah. Okay? Maybe next time.